Good day everyone. I will discuss about meiosis, meiotic nondisjunction and its consequences. Let's first know what is nondisjunction. Nondisjunction is the failure of chromosome pairs to separate during meiosis stage 1 or meiosis stage 2, specifically in the anaphase. In meiotic nondisjunction may occur during either the 1 or 2 meiotic division. If nondisjunction occur during first meiotic division, then all the sperm derived from that primary spermatocyte will be abnormal. In humans, for example, this sperm will end up with a total of 22 or 24 chromosomes, rather than the usual 23. In that figure, meiosis, each gamete has one copy of each chromosome. The nondisjunction occurs when homologous chromosome or the meiosis 1 or sister chromatids or the meiosis 2 is failed to separate during meiosis. If nondisjunction occurs in secondary spermatocyte undergoing meiosis 2, then only 2 of the 4 sperm will be abnormal. Next, we discuss about the errors of meiosis, the chromosomal abnormalities. The chromosomal abnormalities is the incorrect number of chromosomes. The nondisjunction is the chromosomes don't separate properly during meiosis. So the breakage of chromosomes are deletion, duplication, inversion, and the translocation. The nondisjunction is the baby has wrong chromosome number. The trisomy is the cell have 3 copies of chromosome, while the monosomy is the cell have only 1 copy of chromosome. Nondisjunction is the problems with meiotic spindle cause errors in the daughter cells. So the homologous chromosomes do not separate properly during meiosis 1, while the sister chromatids fail to separate during meiosis 2, and too many or too few chromosome. This is the figure in the alteration of chromosome number. At the left side, all the letter A is the nondisjunction of homologous chromosome in meiosis 1, while in the right side, letter B, the nondisjunction of sister chromatids in meiosis 2. In human chromosome disorders, the high frequency in humans is the most embryos are spontaneously aborted. Next is the alteration are too disastrous. And the third is developmental problems result from biochemical imbalance, while the imbalance in regulatory molecules is the hormones, transcription factors, and the second human chromosome disorders is the certain conditions are tolerated, the upset the balance less or the survivable, but characteristic set of symptoms are the syndrome. So the first syndrome that we will discuss is the Down syndrome. Trisomy is 21, 3 copies of chromosomes 21, 1 in 700 children born in US, and chromosome 21 is the smallest human chromosome, but still severe effects. So the frequency of Down syndromes correlates with the age of the mother. So this is the Down syndrome and the age of a mother. So the genetic testing is the amniocentesis in second trimester, the sample of embryo cells, stain and photograph chromosome, and the analysis of karyotype. Sex chromosomes abnormalities, human development for tolerant of wrong numbers in sex chromosome, but produces a variety of distinct syndromes in humans. XXY or the Klinefelter syndrome male, XXX, trisomy X female, XYY, Jacobs syndrome male, and the XO, Turner syndrome female. Let's note the inheritance and the characteristic of each syndrome. So first is the Klinefelter syndrome, XXY male. 1 in every 2,000 live births have male sex organs but are sterile. Feminine characteristics, some breast development, lack of facial hair, tall, normal intelligence. Next is Jacobs syndrome male, XYY males, 1 in 1,000 live male births, extra Y chromosome, slightly taller than the average, more active, normal intelligence, slight learning disabilities, delayed emotional maturity, normal sexual development. Trisomy, or the XXX, 1 in every 2,000 live births, produces healthy females. Why? Because the bare body's all but one X chromosome is inactive. The third is the Turner syndrome, monosomy X or X0. So 1 in every 5,000 births, varied degree of effects, web neck, short stature, and sterile. The genomic imprinting is the phenotypic effects of some mammalian genes depend on whatever they were inherited from the mother or the father. So thus depend on which parent passed along the alleles for those traits. So the genes involved are not sex-linked and many or may not lie on the X chromosome. So the Prader-Wills syndrome and the Angelman syndrome, due to the same cause, are a deletion of the specific segment of chromosome 15. So the Prader-Wills syndrome is characterized by the mental retardation, obesity, short stature, and unusually small hands and feet. So these individuals inherit the abnormal chromosome from their father. So in the Angelman syndrome, exhibit spontaneous slaughter, jerky movements, and the other motor and the mental symptoms. This is inherited from the mother. The difference between the disorder is due to the genomic imprinting. The imprinting status of given genes depends on whether the genes residence in a female or male. So the methyl groups are added to the cytosine nucleotides on one of the alleles. So heavily, the methylated genes are turned off. The animal uses to the allele that is not imprinted. So the several hundred mammalian genes, many critical for the development, may be subject to imprinting, or the imprinting is critical for normal development. In the new generation, both maternal and paternal imprints are apparently erased in the gametes producing cells. Then all the chromosomes are re-imprinted according to the sex of the individual in which they reside. The last topic that we will discuss for today is about the discovery and the characterization of maturation promoting factor. Maturation promoting factor is the famous example of how cyclins and cyclin-dependent kinases work together to control the cell cycle transitions. Is that maturation a promoting factor? The same dates back to the 1970s when researchers found that the cell in M phase contain an unknown factor that could force frog egg cells. So it is stuck in the gap phase 2 to enter the M phase. So this mystery molecule called the maturation promoting factor was discovered in the 1980s to be a cyclin-dependent kinase bound to its M-cyclin partner. So the maturation promoting factor complexes add phosphate tags to several different proteins in the nuclear envelope, resulting in its breakdown or a key event of early M phase, and also activate the targets that promote chromosome condensation and other M phase events. So the role of maturation promoting factor in nuclear envelope breakdown is shown in simplified form in the diagram below. The maturation promoting factor complexes add phosphate tags to several different proteins in the nuclear envelope, so resulting in its breakdown or a key event of early M phase, and also activate targets that promote chromosome condensation and the other M phase event. And that's all my report. Thank you for listening.